So good morning, everyone. I'm Jérôme Boulanger from the Light Microscopy Facility. And I will talk today about image processing for light microscopy. So those who came for EM, image analysis, they can still time to leave. I will not talk about that. So the objective of the presentation is to understand why and when we need to process images, how to achieve a good analysis, what are the steps. We try to understand what are our picture, how can we represent them, how can we understand what we are looking at. And I will try to go through a few core concepts. So it's very high-level kind of presentation. I will not go into detail. It's just basically to kind of acquire some vocabulary and to know where to look for. If you start to Google, you need to have the keyword at least. So this is at least the goal of the presentation. So why do we process images? So we want first is to improve the image quality. So our images are noisy, so we want to improve the signal-to-noise ratio. We can also improve the sharpness. Sharpness is to try to improve the amount of signal versus the noise across the frequency spectrum. We can think about background correction when we have some uneven illumination or some autofluorescence, for example. We can try to estimate our reference frame and to register our image or stabilize the frame across time or across Z planes and so on and so forth. And then, so we end up at this level. What we want to do now is to kind of, at each pixel in the picture, what we could want to do is to attribute a semantic class. So it's what we call semantic segmentation. So we attribute an identity of each pixel. We can give them a color. And the next step is trying to identify who is who. So we can give identity to each of the objects. So it's called instance segmentation in literature, or detection as well. And then further, what we really want to do is to quantify the process we are trying to understand, to study. So in the first place, you want to, the first reason you want to take picture on the microscope. So you want to measure fluorescence intensity, relative intensity, by recovery over time. You want to count the number of cells, of organelles, of foci, their shape. So you could think of area, perimeter, volumes, roughness, or whatever shape descriptor you could imagine. And you could also think of how to characterize a motion you have been able to capture with tracking method, for example. So the goal in the end, often, most of what I see on every day is to try to characterize a phenotype. So we would have, for example, two conditions. So each condition has a corresponding set of images. We can make a list. For each of these pictures, we'll collect some measurement. So we maybe laser pointer. So some measurement here, which we will add, and then further down the line, we will be able to just have this nice kind of statistical interpretation of our measurement and conclude something on our phenotype or what's happening in our data. So this is a kind of workflow where we want to go from our data to some quantitative information, statistically relevant information. So one reason I put this slide is that basically what we are doing is not trying to improve your image to make them look better. It's not Photoshop. It's not trying to make a nice picture for the print. It's beyond that. So what makes a good workflow is we want our workflow to be objective. So what does it mean by that? It doesn't depend on the person who does analysis. So you want to be really independent from your input. You want to be quantitative. So you want to provide measurements which are related to your data, to your experiment. You want it to be reproducible, so you want to be able to run the same experiment again and again and lead to the same conclusion. You want to be scalable, so to have enough numbers to be able to put some stars here, basically. So this is basically creating a workflow, and like this, we could just start and go define our analysis. But often, I find it good to stop before and try to have a look, basically to look at the picture and try to visually grasp an idea of what's going on, because visually you can capture a lot and decide already if there is a chance of getting any information. So often I got, it happened sometimes to me that we want straight analyze with very complicated and sophisticated workflow. And at the end, we had to go back and try to build an overview picture, basically, and try to understand what's going on. Why don't I get the result? Actually, my picture were crap from the beginning. I never looked at them, and it will never have worked. So that's an important step. Just try to skim through your data at least once. So yeah, try guess estimate variability, try to understand what's going on. Discard outliers, overexposed sample, dead cell, etc. So this is, you would say, okay, this is not objective, but actually you have to do this work of not inputting some data you know they are wrong. So this is important. So don't blindly batch process thousands of images if you don't have at least you cannot go and grasp the complexity of thousands of images, but at least have a few examples and go through them. And try to have a way to go from this box plot to the data you have acquired. So to say, okay, this is an outlier, what's going on here? And it's not easy. It's not easy. So there are things you can help to improve this kind of, so the analysis, basically, the image analysis beforehand, before starting any kind of processing. Particular is to add what we call in the team housekeeping labels. Labeling nuclei or cell membrane will enable you to have statistics at the cell level. So if you don't do this, there is very little chance you will be able to collect measurement cell by cell, unless you go manually to do it. You can control the cell confluence level when you do cell on the cover slip, monolayer of cell. It will help a lot to not have measurement coming from one cell to the other. Choose the right microscopy approach. So you have to go and talk to Nick, Sarah, and Tomasz. Optimize your acquisition parameter, your objective, your exposure time. Take care of the spectral overlap if you want to do some colocalization. Think of the analysis basically when you are on the microscope. And think how much my data need to be, what kind of quality needs, or the minimum quality I need to be able to extract the information. You don't need a super resolution image if you are going to just measure the area of your nuclei, for example. So you have to think, it's not to optimize maximum, it's just to tune your imaging to your experiment so that you can analyze something afterwards. So this is, go and talk to my colleagues. And another thing, sometimes we can have analysis where a single little step can be solved manually, and you can basically solve a lot of time just because you are good at finding it, and finding an automated method to do that will take you a week, but maybe in an hour you could have done it with a small interactive step. Okay. So you have to find out the balance between what to automate and what can be manually therefore. So what's an image? It's simply a measurement. So you're really measuring something on a regularly sampled grid. So at each pixel here, each coordinate x and y, or row and column, you will have a value which is a measurement. So we can have a number. So this representation is just an array of pixels. So we can represent this array also in different way. We can represent it as a continuous function. Like here, I represent like the z-axis here will be of this landscape, will be the elevation of this landscape is exactly this. So the brightness of the image. And then you can think this would be a sort of a function in on 2D space with a which has been sampled at different points. So then you can use different kind of tools. You can also represent this as a sum of little sine and cosine function at different frequency, and look at it as a Fourier transform. You can think each pixel is actually a node in a graph with some link to the neighboring pixels. So it would have a kind of very connected graph. This is with a random value and probability of transition between the pixels. So it's a random field, an Ising model, for example. You could represent it, want to localize the frequency content. So you can think in terms of wavelet or scale space. You can represent little blocks of the image as a dictionary of features, or you can embed this matrix in a high-dimensional space with many features to describe this picture. So image filtering, we are here. So the basic steps you can do on an image is to combine the information from neighboring pixel, like here in this orange block, and take a function of all these pixels and replace the middle value, not replace, but save the middle value into a new image. So for example, you could do the average, you could compute a median, you could do a standard deviation or whatever. A Gaussian filter will be defined here as well. So this is a linear filter as long as the combination of the pixels is linear. So a median filter will fall in this category. So you would have the median, the minimum of this block, the maximum filter. So this start to look like this morphological operation you can do, opening and closing, which kind of extend or shrink the borders, the edges on the image. Yeah, a linear filter, an interesting thing. So it can be also called a convolution, and we will see that a convolution is also a multiplication in Fourier space. So this convolution is actually using a sort of brush to replace every line we will see in the image. So this brush is called a filter or a kernel. And the operation of using this brush to represent this line, to paint this line, is called a convolution. So there are different frameworks. So here I will just talk about this kind of approaches where you have some data, the data you have collected, called Y here, and you would have an estimate here, X. And you would like to know, basically, we'll find the estimate which maximize my maximum a posteriori. The maximum a posteriori is based on the likelihood, so the probability of observing the data, knowing the estimate, and an a priori, so some smoothness that you give, or some other information. The smoothness is a typical thing, which is here. So you will combine these two information to define a cost function. And this kind of iterative algorithm can help you basically minimize this cost and in meanwhile estimate the X and recover, for example, a sharp image. You could denoise an image, noise-free, create a segmentation, estimate the motion, etc., etc. So this kind of framework also kind of encompass even linear filter. You can express a linear filter as performing this kind of operation, or yeah. So it's not always necessary to do a gradient descent like I do here to minimize this cost. You can have an like closed form solution. There are some other techniques where which are learning based. So here we will collect some input and some output which are known. So either you manually define them, so set the label, for example, manually, and you will define a link between the input and the output with a free parameter W here that you want to estimate. I put W for weights, like it's usually in neural network now. And then we define a loss function, which will minimize, which we will try to minimize, which makes a link between our prediction here and the actual labels which are known. And then the next time you will see X, you will be able to use a W you have estimated, or the parameter, to predict a correct Y. So these are used for classification. The basic technique application will be a classification. Like here, we find that this is a cat, but we could do also the same learning-based approach for segmentation or detection. So at each pixel, we look at the pixel and we decide, is it not a cat, but is it foreground or background, or et cetera, et cetera. You could do segmentation of a cat, but we don't have too many cats in our microscope, fortunately. So one first thing I mentioned at the beginning of the talk is that the image we take, they are noisy. So we are often, by design, in a photon limited imaging, at least in fluorescence microscopy. So the number of photons are a function of the exposure and illumination. The photons interact with the sample, so they create free radicals, they bleach our fluorophore. We don't want that, so we have a limited number of photons we can use to probe our sample. So we want to be careful with that. And then here we can see how the image changed from a long exposure time, from 1.5 seconds to 10 milliseconds on the same kind of sample. So we see that the image basically becomes quickly very noisy. So the noise source are, we have three. So we have, the first one is intrinsic noise, so linked to the fact we are counting photons, which are few. So this has a Poisson distribution, which means the strength of the noise depends on the signal. So the more there is signal, the more there is noise. Let's say the signal to noise ratio is still improving. Okay. Then there is a dark current, which is a number of photoelectrons which are generated even if there is no signal. So it's the number of electrons per second. In nowadays instrument, this is very small, especially at the exposure time we are using. And the other significant noise is a redox noise. So this is more for camera-based system, not for confocal. So we have thermal fluctuation in electronic circuitry, which lead to variation of current and which pollute our noise. So this is of the order of 1 or 2 electrons. So we can characterize our sensor. We have a QE, which gives us how many photons will be converted to a photoelectron. We would have our readout noise here. And then some other characteristics such as the frame rate. But this data basically will give you the noise distribution and allow you to compare sensors. So now we might want to remove this noise or try to reduce it. So now we understand what is noise. It's a random fluctuation of 1 pixel. So it's not, here we have a very specific kind of semantic. We don't say noise is all we don't want. It's just this kind of noise. So here, if we want to think what will be denoising, so it will be removing and trying this random fluctuation, which could be seen as estimating an average of many similar images, which would have been taken of the same scene. This average will naturally decrease the noise as the strength of the noise over the number of images. And so what we want to do is to remove the stochastic noise while preserving the fine feature and the intensity. So we want basically to try to estimate this ideal image, which would have been taken, for example, with a long exposure time or on better condition. So in the past, I've proposed this method, which is a non-local patch-based approach, which is able to take into account the two kind of noise contribution, the intrinsic noise and the readout noise, and which basically use this a priori, which image blocks are similar to a certain point, and does this in 3D on time. Virtually, all this denoising approach could be seen with this kind of diagram where you have an exposure time, and you start at 20 milliseconds. So this is before you have your, if you have a reference image, you can measure a kind of PSNR, a quality metric. And then after denoising, you will jump to this point on the curve, which you could map to the original quality metric of the image, and get a virtually corresponding exposure time. Let's kind of illustrate a bit more concretely the gain in exposure time from 20 milliseconds to 300 milliseconds. Another popular approach based on the neural network. So you need a training of a pair of noisy and noise-free image. You train a unit neural network to map the noisy image to a clean image, and then you will get this kind of mapping. So this can go beyond image denoising, can also improve sectioning or of sampling of the image. There is also some version of it where you don't need this clean image, you will need only a noisy image, and using the fact that the noise is independent from one location to another, you will be able to recover and train directly on the noisy image, the denoiser. Another aspect of the data we have is that the data are band limited. It means that there are a limited number of frequencies which go through the microscope. So we saw this on Tuesday with Nick, that there is a cutoff frequency on our microscope, there is a resolution limit, and beyond this, we don't see anything. I'll try to illustrate this a bit. So basically, we want to approximate our what we see, our image formation, and we have a 3D linear filtering by the point spread function, what we call the blur of the objective is the point spread function. This linear filtering is also, as we said already, a convolution, and it corresponds to a multiplication in Fourier space at what we call the optical transfer function. This optical transfer function vanishes beyond the Abbe resolution limit, which is lambda over 2NA. So here we have this figure where I drew this circle which corresponds to our resolution limit. And in a beautiful, perfect picture like this without noise, there will be only zeros here. Everything which is beyond this, this line will be only noise. So in a normal situation here, you will see some gray, a bit like this. but it won't be just, it would be just information, not just noise, not information like we have here. So a result of this resolution limits is that, as we already saw, that Nyquist-Shannon theorem says that you don't need to sample at twice this resolution limit, so more than twice. So the ideal sampling will be lambda over 4NA. So yeah, this theorem which says that if you have a band-limited signal, it's sufficient to sample at twice the bandwidth. So here is an example. So we have a sample. We have two samples here. One has little stripe in this direction, and one has stripe in this direction. So we can see in the frequency domain that the position of these blobs here correspond to the orientation of these stripes. Here, it corresponds to these stripes. Okay. Now, a low-pass filtering will crop here everything which is beyond the cutoff frequency. So it's quite small here, but compromise for visualization purpose. Basically, we lose these little dots here. And when we look in the microscope, these two samples look identical. Okay, so that's what microscope does to you. You lose these details, and you don't know, you can't know. If I give you these two pictures, you don't know which is which, basically. So you lose this information. You can look at this in a more like relatable kind of picture. So we have this acting circling of the axon, which have been imaged using a super resolution microscopy technique, which Sarah will present next week. And then if we look at what it should appear in a wide field microscope, or it will, so here we see, we can look at our orientation. So this frequency here, which are very pronounced, correspond to these rings. Our microscope will clip only the inside, and we will see only this. We can't see the rings. So again, it's the same idea. So what is exactly this blur? So in 3D, it's a 3D function called the point spread function. It models the microscope. It's sort of model of what you see. It summarizes all the imaging process in one picture. So we can it's actually a picture. So we still have, it's a model, so it is not perfect. It assumes that the response is linear on the same, so stationary across the whole field of view. And it is just the image of a point viewed by the microscope. So it's basically the brush. So this is our brush for here, confocal at 1 area unit and a confocal at 5 area unit, which basically becomes similar to a wide field. It's function of the numerical aperture, the wavelength, and aberration. It's not a function of the magnification, for example. So, I mean, it would be just scaling it, but not changing its shape. So the idea to invert this model, the idea is to recover a sharper image, taking into account the knowledge we have on the image formation, which is this point spread function. So the idea is to inverse the convolution operation, inverse the brush, recover the sharp line, and it's called deconvolution then. So we want, with this, to increase the sharpness on the signal-to-noise ratio. We saw that in Fourier space, a convolution will be just a multiplication. So we could go ahead, divide by our optical transfer function, and recover a sharp image. It's almost good, except we saw that the optical transfer function goes to 0 beyond the cutoff frequency. So not good. It also goes down quickly, so it will amplify the noise. You will start to multiply by very small values of the OTF, and the noise also depends on the intensity. So you have a very complex problem. You can use Bayesian approach with a noise model and a priori, like I presented earlier, to prevent noise amplification and optimize this cost function. You can have, from this setting, and if you assume instead of a Poisson noise, just a Gaussian noise, you would have a least square approach, and you can derive directly a linear filter, which is a linear filter, which you can apply in one step. This filter is what you see in the software, for example, in the Zen software when you do area scan, or in various other situations. I think you will see this name appearing. You can have an iterative method, which is tailored for Poisson noise, so called the Richardson-Lucy approach algorithm. And you could also use a standard optimization algorithm to optimize your cost function. So like the conjugate gradient, which is used in Huygens, for example, which we have in the facility. So here you get from a blurry image to a sharper image. You can start to, so you have interpreted your image based on your prior knowledge of the image formation model and the smoothness of the image as well. So yeah, we have several options here in the facility. You can use Huygens. You can use, there is modules in Nikon Element, for example, on the iSIM. And then in the area scan, I think the Leica software also might have something. In Imagier, you would have Deconvolution Lab, and you can use PSF generator. You have Ops in Fiji. You can use MATLAB or Python. There's a lot of options where you can use this technique. Once you have noise-free and sharp image, you might want to relate several pictures together. So this is image registration, trying to align the pixel of two images. You can have different situations where you need to do this. For example, if you have two channels, you might have a misalignment of the channel. You can have a drift over time, or you would like to estimate some force in the image or do some tracking. You can do image registration is necessary, required for optimizing stitching when you have a large set of tiles. And then maybe you can also register your image to an atlas, so a known reference of your organ, for example. So the input data for registration can be two kinds. It can be some landmark, so control point you have manually set in the image, or which have been automatically detected as what we call interest point. Or the registration could also be intensity-based, using directly the pixel information to align the two images. Or it can be also both in some other context. So for representing this deformation or some function which will map the two images, you can have some different kind of model. You have parametric model, like a translation, rotation, a rigid body, so combination of these two, or an affine motion, etc., etc. Or you can have a non-parametric model where at each pixel you would have a deformation field. So you would have many. So this is a lot of data. This is very compact and way to represent deformation. So depending on what you expect has happened to your image, you would have to choose one of these two model approach, one of these two kind of input, and then go for the algorithm which work for these two things. So in terms of algorithm, there are different approaches. You can have simple correlation. So this is intensity based, where so there is a link. The basic thing is that there is a link between, so this correlation-based approach is often performed in Fourier space because there is a link between translation and the Fourier space. So, so we can use cross correlation to recover our shifts. So here it will be only for translation in general. So phase correlation, for example. You can have also an idea of rotation and scaling using what is called a Fourier-Malin transform, which transforms the image in a polar geometry and apply the phase correlation on this transform image. You can also use correlation-based correlation-based approaches to estimate the dense, so non-parametric displacement using here. So in each little block, we will look what is the most similar block in a neighborhood. So using normalized cross correlation, for example, this often doesn't impose any smoothness on the result of the vector field you are estimating, or displacement field. So it's often very noisy. It's just some, so the smoothness will be linked to the overlap of the patches of the blocks you are trying to find. So the bigger the blocks, the more regular the result will be. The smaller, the more noisier, but more precise at detecting small distortion. In landmark-based registration, so you need to find corresponding points unless you already provide them as being pair. So you can use different algorithms such as the Ransack, which is a robust estimation of the deformation matrix and the matching of the point, or this method called coherent point drift method, which also does this. It basically it estimates, yeah, in the least square approach in a robust way here, the link between the point and the deformation. You can have also a non-parametric approach where which is called a thin plate spline, which will interpolate the deformation to best match the pairs. You have a software such as the Big Warp in Fiji, which implement the thin plate spline. In Python, MATLAB, you have PySciPD or computer vision toolbox. In ITK, you would have both approaches. So landmark-based registration was often used also to create 3D scenes. So it's a lot in computer vision and also in medical image processing. So when we want to estimate a flow or a deformation field at each point, we call this we can call this optical flow or particle image velocimetry. So we can assume the preservation of intensity over time. I want to estimate the displacement, as I say, at each pixel. There is an ambiguity often along a contour because you not know if this point here went here or it went there, because it's the same along this line. So if we look at this pixel, it could be anywhere along the line because they look similar. So for improving this, normally you have some smoothness, which will use information across these corners to propagate it where the information is less rich on the line. You can use, again, this block matching approach which is linked to the correlation. It will be pretty noisy. You can use a priori to smooth, and often this technique uses a multi-scale approach. So you downsample this image, get first estimate, and then refine by going back to the original resolution. A next topic is spot and blob detection. So here we have the blob is anything which is more or less isotropic without any specific feature. And often in microscopy, we have a lot of blobs. So because we have a lot of objects which are non-resolved, very small, not any specific shape. So blob detection happens often. So there are different ways of implementing or a blob detector. So you can look at the local maxima, the brightness spot. Here would not work because the blob are dark, but local minima will probably do something. But then often you need to pre-process your data with some sort of filtering. And this filtering will give you the scale at which you want to extract the blob. So when you use like a difference of Gaussian, the sigma parameter of the difference of Gaussian will give you the scale at which you find these spots. So there is also ways to not define in advance, to use different Gaussian function and try to find at which scale the spots are. So because spot could be at different scale. So this is all integrated in like a space case theory, and the same for the Laplacian of Gaussian can have different scale. Basically, the Gaussian filter will give you a scale in the image, like a resolution. So you can then extract this blob at specified scale or a multiple scale if you test for several scales. It's an easy way to detect any kind of vesicle, nuclei, etc., when you don't need something more sophisticated. So it's an easy first try. And you can combine it with Gaussian fitting, for example, to enable super resolution, for example, in single molecule localization microscopy. And it's very easy to script, even in ImageGA macro or MATLAB or in Python. It's implemented in TrackMate as a first step for detecting the object, on IEC as well. IMAIS doesn't say how they do it, but there is a spot detection module in IMAIS as well. So another task we can try to tackle is segment our image. So to assign a class to each pixel. So we are here in a semantic segmentation. We just want to give a class, or in instance segmentation, we want to identify who is who. So find nuclei 1, nuclei 2, nuclei 3, cell 1, cell 2, cell 3. So this is instance segmentation. Our basic approach, like a thresholding, with maybe combined with some filtering. You can use watershed, which will try to separate touching objects. You can use more advanced techniques such as active contour, or you can have snake level set or balloons, which are also implemented in Fiji. You have great graph-based method. So you have Markov random field, which is considering the image as a graph, and graph cut is a way to find the partition in the image which minimize a flow between the source and a sink, which it can be used as a post-processing of, for example, a neural network will give you a probability of belonging to the object. And then, so you have all these machine learning approaches where you could have pixel classifier, pixel classification, where you train, give example of pixels which belong to different class, and then the classifier will extract some features. So apply different filters to the image, and at each pixel try to decide to which class the new pixel you didn't label belongs to. You can use UNet in the same idea as you have some example of pixel, and then the UNet will try to predict the class, and you can have some more advanced technique which try to perform so instance segmentation, basically to separate the objects which are in the image. So all these basic methods, these are everywhere. These are a bit more niche, and then this becomes much more popular. So here we have pixel classification. I would recommend LabKit, for example, in Fiji, which has a starting point. And then all this deep learning approach can be a bit more involved unless they are already pre-trained, like StarDist, very good for finding nuclei or cell pose for segmenting individual cells. So you can go for it. It's easy to use. if you don't have a pre-trained network, this requires a lot of more data than pixel classifiers, so more effort on your side, and you need to justify it for yourself. You need to spend time annotating. So StarDist as a simple go-to method for nuclei segmentation. Well, it's not so simple, but it's available at least. It's easy to use, for example, in Fiji. So how does it work? It represents the region of interest with a star convex polygon. And it learns for each pixel the distance to the boundary in a k direction. So for each pixel, there will be a distance to the boundary. So for example, this at each pixel, you would have different orientation, which are scan, which are a parameter of the algorithm. For example, often they use 32. So at each pixel, we'll have 32 values, which are the distance to the boundary. And then for the direction, the distance to the boundary in this direction, and then an Euclidean distance to the boundary, which can be used to as a post-processing here to find the instance, basically, so to distinguish these three attaching nuclei. So basically, you have a network which, from the image, will predict these 32 distance along a radius, direction, and then an Euclidean distance to the boundary, wherever the direction is. The architecture is a unit, so this kind of constricting loop and expanding loop. So an encoder, decoder with some skip connection across the different resolution. For cell segmentation, for now, CellPose is very efficient. So it's another approach which can use two channels for cell segmentation. We could use the nuclear channel on the membrane labeling, for example, or cytosolic labeling. So what it does, it's again a unit, but the input will be these two channels, not only one channel. The output will be three images. You can see three. So at each pixel, we will have three values. We would have a flow and a probability of being inside a cell. The flow is basically telling you where to go if you want to find the center of the cell. So how did they compute this initially, the flow, when they did the annotation? So you have the mask here, you have a cell, you get a mask. Then at the center of the cell, which is defined in various ways, so you can say the barycenter of the cell here, they will diffuse the information from the center using a heat equation, and then compute the gradient of this heat equation, which are vertical and horizontal, give you this coding, for example, which gives you like a direction where to go. If you are here in the corner, you want how to go to the center if you backtrack it. And then, yeah, basically you have for each image and these two flows plus the inside-outside probability, which you can use to find the cell. So the output is really this three image, and there is a post-processing which backtracking the flows. So it has been trained on many pictures, which makes it with different. There are different models, and it has been also improved over time. So the CellPose 2 introduced the human in the loop, so it's easier to. If it doesn't work for you, you can annotate a few images and improve the model to match your data. So this is very convenient. And CellPose 3 introduce also image restoration along to help the segmentation. So once we have objects, maybe we want to track them over time. So tracking here means maintaining the identity of objects over time. So if one cell A in one picture, it becomes cell A in the next picture, because originally we don't know which one is which. You just have masks. They can have different label after segmentation. So the difficulty when you do tracking is the density. So there's two main parameters: the density of the object, or the in the feature space. So it could be, for example, you are big and small moving, you know that the big goes to the big and the small to the small. So it's density in feature space. So if they are all small and all at the same place, this is difficult. And if they move a lot, so that's the other thing. So if the tracking doesn't work, you should work on these 2 points. So density probably is hard to control. Apparent velocity, you can improve the frame rate if you don't manage to track. Basic algorithm, there is nearest neighbor, you minimize the association, or you minimize an association cost. So there is two approach. Basically, find the nearest one and say this one is my next identity, or you consider the whole point cloud of a 2 time point and you try to match them, minimizing the global association cost. Another approach is which can be combined with this, is to have a motion model to predict the next position. So you basically say I've been going this direction before. Next step, I go this direction. So I predict I'm going there, and then you minimize the cost matrix, the distance between the prediction on the measured one. So this is the next frame. The position in the next frame. So this is what you can do with filtering. So there are challenges and particle tracking challenges which list a lot of the techniques which can be a starting point for knowing what software to use. We recommend TrackMate as a starting point, the easiest tracking algorithm which is available. So in Fiji, in IC there is a very sophisticated multi-hypothesis tracking algorithm, which is I never managed to use it really, but it's on paper. Very nice. MRIs also has very convenient 3D tracking with visualization. Nikon Element also have. In Python, you can use TrackPy, LabTrack, or BTrack, for example. And then you can move on having this track to analyzing and understanding what kind of information you have. For example, you can measure instant velocity. You can measure simply the variation of feature of intensity over time for each object. You can compute mean square displacement to have an idea of diffusion or subdiffusion or active transport, for example, having different kind of mean square displacement curve. In practice, mean square displacement is very difficult to obtain in microscopy data because you need a lot of track. It's like an ensemble measurement, so you need a lot of data. It works when you have a lot of track on long tracks. So this is a bit hard. And then once you have all your track and you want to go like armor track changing kind of motion model over time, so you can have a multi-state model for the motion, so where you have some state which correspond to different diffusion coefficient or velocities here. And then you have hidden Markov model, which says, okay, I have a probability of changing from one state to another one, and this will be estimated in the same time. So that this transition probability are estimated at the same time on this parameter. So, and then you can classify the kind of motion you have in your cell. I think almost finished. I will talk a bit about colocalization. So here we want to measure the interaction between two labels. So fluorescence microscopy is where one big advantage, you can have different channels and which correspond to different protein, which give you their localization in space, and you can give an idea of if they are the same place up to a point. So you could have a correlation approach. So an intensity base, are my two signals correlated? Are they on the line like here? So we'll have high correlation. Or are they not correlated using this? So here we have red intensity and green intensity. So we see here they are correlated, but here they are not. So they're not on this line. You could have a more region-based, like an overlap coefficient, where you try to measure the amount of signal in this intersection versus the signal in this red segmented region or green segmented region. So you can compute these two ratios, the Manders coefficients. You can also look at the distance between objects. So this is an object-based co-localization, where you look how many objects in one channel are, so this one in this ring are close to this object in the other channel, and you create this percolation G function, which, if properly normalized, should be 1 if there is no, equal to 1 if there is no interaction. And it will give you an idea of here to scale the distance at which the particle interact. So then there is a question, how much equalization is due to randomness? So you see here, this is number 1 is our randomness. Here is a bit more, and here is a bit more complicated. So you need some randomization test. You can randomize your image and recompute the coefficient to get a p-value. I think the most interesting thing is to have experimental repeats. So you can go through several measurements and compute a p-value based on that. And finally, a last thing is what you might want to do is to build up hierarchy of your mask or segmentation based on proximity or inclusion. So you can have several semantic levels for this. You can have the image, part of an organ, for example, cell and organelle. So how many Golgi stack I have per cell, for example, this kind of thing. You can use basically mask and logical tests. So it's very, can be if you program it, quite easy to do. You can use distance transform for testing proximity. We have like this kind of analysis is very easy in Nikon Element using the GS3 module. You can manipulate ROI in Fiji. It's easy also to test if one ROI is inside another one. You have a cell biology module in Imaris. In QPath also you can, by default, you need to define a hierarchy. And then, yeah, you can use region props in scikit image, for example, to test inclusion. So just some quick list of summary of a tool we kind of have in the facility or elsewhere. So we have Fiji for general tool, Imaris for visualization, 3D visualization, spot detection and tracking. So rendering is very good in there. You can do a nice movie. We have 3 license. You have GS3. We have 2 license. It's easy to define graphical workflow. Huygens is a reference software for deconvolution. We have 2 license for deconvolution, and also they include colocalization. Napari is an emerging, more and more emerging tool for visualization and interactive processing, a bit like Imaris. We have Appear for online workflows, so now it became Zeiss something. Cell Profiler for screening workflow. IC is also a general tool, which was planned to be a kind of a sort of replacement, equivalent to Fiji. Elastic for machine learning. We use a lot. We help you use Python and scikit image, for example, for creating scripts. MATLAB also, we have a few license in the analysis room. And we are also preparing a course on QPath for boot slide imaging. So at the end of next month, we will have a training on this, which I'll just maybe skip this and go directly here. So for assistance on training, you can come and talk to us. So don't, yeah, just come. So we have 3 image analysts. We can help you to advise you or help you write code. And then we also propose several training. So scientific imaging with Fiji to have a more hand-on approach, how to script using imaging macro, how to define workflow using ImageJ. We have a Python-based course, Oregon's course by Dina. QPAS is coming with CMENG, and we also propose GS3 training sometime. So just look for them when we announce them, and then thank you for your attention.